values in the life cycle analysis of any product. So it's always better to have some plan of action or some strategies so that we can reduce these inhibitor formation initially, or if at all they are present, then how can we mitigate if they are present. So there are multiple ways we can think about reducing the effect of these inhibitors and or the production of these inhibitors during the pre-treatment. So that will be discussed by Dr. Jitendra Kumar Sani. So thank you, Dr. Jitendra. And just to briefly introduce Dr. Jitendra, he has been working in this particular area on renewable energy, lignocellulosic biorefinery, and enzyme microbial technology for last more than 10 years. And he started his career as a research associate in Department of Animal Nutrition, Gadwasu, Ludhiana, and then followed by junior and senior scientific officer at DBT Indian Oil Corporation at Center of Advanced Bioenergy Research, Faridabad, who are working on this particular field. That particular center is working on this field. And then 2016, he joined Central University of Haryana as assistant professor, and his MSc and PhD from MSc from Gurukul Kangadi University, Haridwar, and PhD from JB Pant University of Agriculture Technology, Pantnagar. And he has, to his credit, around 40 research publications, which have got more than 2800 citation and H index of 23 and I10 index. Was able to publish very good research papers in very reputed journals, including renewable energy, bioresource technology, and industrial crop research and product. So a very good mentor, very good researcher, and he is also member of scientific societies like AMI and BRSI and Asian Federation of Biotechnologists, and has got many awards and accolades to his credit. So with this brief introduction, I invite Dr. Jitendra for talk.
So, good afternoon to everyone. In today's lecture, I will be basically discussing about the inhibitors and what are the ways by which these inhibitors are produced and how we can mitigate these inhibitors basically. Okay. So, in lignocellulosic biorefinery, we have different operations and during different operations we can get different inhibitors. Okay, and these inhibitors are not only the today I will be discussing about the biorefinery process in brief. What is the biorefinery process? How do we do? What are the different steps? Okay, what are the substrate for biorefinery? Because to understand how these inhibitors are generated or from where these inhibitors are coming, so first we need to have basic idea about the biorefinery process. Then I will be talking about these different types of inhibitors and their mitigation mechanism. Okay. Thereafter, I will discuss one of the strategies. There are lot many strategies which we can employ to mitigate these inhibitors. So we have worked on the one of the strategies. So I will discuss some of the results, and then of course we will discuss. We will have the conclusion and acknowledgement check section. So first, coming to the biorefinery operation. So what is biorefinery? So by this time you must be clear. What is the biorefinery process? Okay, you know. Everybody knows what is biorefinery? And so biorefinery and biofuel industry, they are distinct in the fact that when we talk about the biorefinery, so we are basically dealing with multiple products. Okay, there should be a spectrum of products. Okay, from which we can choose what we actually want to produce. Okay, so we can produce bioethanol, and along with that we can also produce some other sugars or some prebiotic oligosaccharides, or we can generate some acids like succinic acid or citric acid. Okay, so it depends upon the type of process that we are going to use, but the main thing in the biorefinery is that we have to produce multiple products. Okay, so that we can improve the economy of the industry. Okay, so by definition it is a facility that integrates the biomass conversion processes and equipment to produce a spectrum of products. Okay, And these include biofuels, chemicals, materials through various approaches. So we can have different approaches, like one of the approaches to convert these compounds, or you can say the resources which are present in the biomass, into the product using the sugar platform. Okay, so we use sugar platform or thermochemical platform. These are the two different, you can say, technologies by which we can convert the biomass into products. Okay, and this sugar platform is basically the biochemical route in which we use different types of microorganisms or their enzymes to convert the biomass components into the useful product. Whereas in the thermochemical platform, we use the thermochemical processes like pyrolysis, then gasification. Okay, so these type of processes which only employ the thermochemical, you can say, means to convert the biomass into different products. So we can have different types of these platforms. So I will be confining my lecture to the inhibitors that are generated during the biochemical platform, or you can say sugar platform. Okay, and I will be taking the specific example of lignocellulosic biorefinery, and in that also I will be mainly dealing with the bioethanol production, because that is the major product that we deal with in basically the lignocellulosic biorefineries. Okay, so we can also compare this biorefinery, especially the lignocellulosic biorefinery, which is basically based on the lignocellulosic biomass. So these are the feedstocks that are the starting material in the lignocellulosic biorefinery. And we can compare it with the petroleum refinery. Just like petroleum refinery, we use crude oil to convert it into different products like diesel, gasoline, kerosene, naphtha. And in similar manner, we can convert this lignocellulosic biomass, which can be in the form of different uh, resources like agro wastes, like paddy straw, wheat straw, then municipal waste, and then woody woody materials. Okay, so these can be converted into different products like ethanol, lactic acid, xylitol, and succinic acid. So these are some of the representative examples, but there are multiple products which we can produce in the lignocellulosic biorefinery. Okay, so you might be going through this slide again and again, like what are the first generation, second generation, and third generation substrates. Okay, so first generation substrates, they are basically the food materials like grains or sugars, and second generation substrates or feedstocks, they are the agro wastes or lignocellulosic material basically, and we deal basically with this second generation when we are dealing with lignocellulosic biorefinery. So we are dealing with the second generation feedstock. Okay, then of course there are algal feedstocks also that come under the third generation. So now coming to the structure of this lignocellulosic biomass, so it is important to understand the structure because there are three major components basically in the lignocellulose. Okay, lignocellulosic material is the structural material that constitutes the cell wall of the plant. So it is basically present in every plant or uh, tree. Okay, so it is a major component of the tree or the plant material, and it is almost you can say that universally it is the most abundant material, biomaterial available on the earth. And it is a complex that is made up of basically three type of biopolymers, that are cellulose, hemicellulose, and lignin. Okay, but the main thing is that these three components, they are not present in a particular fashion. They are distributed randomly, and they interact in different variety of manners, which give rise to the overall complexity of the biomass. Okay, so overall interaction of these cellulose, hemicellulose, and lignin, it contributes to the overall complexity of the biomass. And you know, the cellulose and hemicellulose, they are the major components which can be converted into useful products. Okay, and for that purpose, lignin basically it acts as a hindrance. So it is often desirable to remove this lignin during the pre-treatment process. Okay, so this slide just shows the like how these different components, they are distributed in the lignocellulosic biomass. So cellulose, it is the major component, and it is present up to 50%. So basically, depending upon the type of biomass, like some could be grassy material, some could be agricultural waste. Okay, then paper waste, waste coming from the textile, and apart from that, woody material also. So they will have different, uh, you can say, the exact composition of these materials will vary. Okay, so there is always a range when you study any paper. So they will give the range, different range in which the cellulose will be present. So like up to 50%, then hemicellulose will be present up to around 30%, and then lignin also it can be present in some of the biomass. It could be very high up to 25 or even more than that. Okay, so this cellulose and hemicellulose, they are basically the polymeric material, and they can be converted into some constituent sugars. Okay, but lignin is complex. It is a aromatic structure, and it is highly energy. It has high energy content, and it is highly recalcitrant. So this lignin is the major portion which has to be, you can say, broken down or removed properly during the pre-treatment process, so that we can convert the biomass into the desirable product. Okay, so this figure just shows the 3D structure, how these lignin, cellulose and hemicellulose, they are interacting with each other. Okay, so you see cellulose, it is present in the form of small, you can say, chain-like material. Okay, so there are chains of this cellulose, which is basically made up of the glucose as a subunit, and then hemicellulose is distributed as a side chain. Okay, it is also deposited, but these chains of hemicellulose, these are extremely short in comparison to cellulose. So we can easily remove this hemicellulose during the pre-treatment step. And then there is lignin. So lignin is deposited here and there, and you can see when the enzyme bind to this cellulose or hemicellulose, and it proceeds for hydrolysis, so lignin will basically act as a hindrance. Okay, it will obstruct the path of the enzyme, so it is extremely important to remove this lignin. So this slide just shows the importance of the characteristics of cellulose. Basically, cellulose is made up of the repeating unit is cellobiose, okay, which is composed of 2 units of glucose. Okay, so then there are non-reducing and reducing end. So reducing end can participate in some chemical reactions. Okay, and it has the property to reduce. It can act as a reducing agent, and in cellulose chain, these different cellobiose unit, okay, they are repeated many times. Okay, and this repetition is basically termed as the dp value, or degree of polymerization. So in case of the cellulose, the degree of polymerization can be up to 10,000 or even more. Okay, but in case of hemicellulose, it is hardly 500, 200, or maximally it can be 100 or 1,000. Okay, so these different chains of the cellulose, when they are synthesized, so they can bond together. There can be weak interactions like hydrogen bonding, and through that hydrogen bonding, these cellulose chains, they are tightly packed. Okay, they are arranged in a highly, you can say, systematic manner. They are arranged parallelly. Okay, and this will give rise to basically the crystalline region of the cellulose. Okay, and apart from that, there are certain regions which are, you can say, they are not highly compact. Okay, and they can be easily broken. So these are the amorphous regions. So cellulose overall, it is composed of this, you can say, the crystalline as well as amorphous regions. Okay. Now hemicellulose, so hemicellulose is the remaining polymeric carbohydrates which are present in the cell wall, that are neither cellulose nor pectin, and they have beta 1,4 linkages. Okay, just like cellulose, they also have beta 1,4 linkages, but the substrate in case of the synthesis of hemicellulose, they are different from cellulose, and they are variety like xyloglucans, xylans, mannans, and glucomannans. Okay, so we have different sugars which constitute the backbone of the hemicellulose. Okay, and when particularly xylose is present, so that will be termed as that homopolymer. That polymer will be termed as xylose. Okay, or xylan basically. Okay? So these different types of sugars, they are present in the hemicellulose structure. You can see hexoses, they are present. Different hexose sugars are present, as well as these pentose sugars. Okay, so variety of these sugars, they are present, and they can, when this hemicellulose is hydrolyzed, either during the enzymatic hydrolysis or during the pre-treatment process when it is broken down, so variety of these sugars, it will be released. Okay, and they can be arranged in different manners like arabinine, galactomannan, xylobiose, xylan. So there are variety of combinations in which these hemicelluloses, they are present. Okay. Now lignin, so lignin is a basically the monomers in case of lignin, they are the phenolic compounds. Okay, and these phenolic compounds, they are basically three types of the you can say the starting material for lignin. Okay, one is the p-comeryl alcohol, then coniferyl alcohol, and sinepyl alcohol. These are the basic constituents of the lignin, and these constituent, they are also termed as H-lignin, G-lignin, and S-lignin. And these lignin, different types of lignin, which contain these different types of monomers in different concentrations. So they will vary in different types of biomass. So depending upon if we are having like G-lignin is high in the biomass, then it basically forms a tight structure and it is difficult to degrade. Okay, but S-lignin on the other hand, it can be easily removed. So if we perform our pre-treatment in such a manner that this ratio is increased, S to G ratio, okay, so that will make the biomass easily amenable to enzymatic hydrolysis. Okay, and when these monomeric units, they interact, so different types of bonding can be there that can occur, and it will lead to ultimate this complex structure. Okay, so that you can see this complexity of the structure in comparison to the cellulose or hemicellulose structure. Okay, so due to this complexity, there are steric hindrances, and the structure is also very compact. So it is very difficult to break down this particular compound into its monomeric units. Okay, so we can get short fragments, we can get different types of products out of this lignin. And another important thing is that lignin is not very homogeneous. Okay, as can be seen clearly in this picture. So there are variety of different constituents, different components. So one time this bond will be broken, another time this bond will be broken. So the product that will be generated out of the lignin degradation, they will be highly variable. Okay. Now coming to the biorefinery concept, so using these different types of lignocellulosic biomass, we can have these different biomasses like corn cob, beechwood, corn stalk, wheat straw, cotton residues, and sugar cane. So these are the some of the representative, you can say, biomasses which are containing high quantity of lignocellulose. Okay, so these lignocellulosic material, they from these different components, cellulose, hemicellulose, and lignin, we can get different varieties of these products. Okay, so we can get a spectrum of products like biofuels. We can categorize these products into different categories like biofuels. Okay, so we can have bioethanol, biomethane, and biohydrogen. Okay, then platform chemicals like xylitol, lactic acid, lipids, sorbitol. Then we can also get biomaterials from these components like biosurfactant, biopolymer, and biopigments. Okay, then lignin can also be converted into phenolic compounds that can be used for binding, dispersing, and as emulsifying agent. Okay, so we can get variety of these products, and depending upon what type of microorganism basically we are going to use in the fermentation process. Okay, so we can use different bacteria, yeast, or molds to convert these components into different products. Okay, now coming to the biorefinery process, so I'm taking the example of bioethanol productions, how this bioethanol is produced using the this biomass as the starting material. So if we take the sugar platform, that is the biochemical route basically. Okay, so we are using the enzymes or the microorganisms to convert these components of the biomass into the final product, which is ethanol here. Okay, so we can divide the operation basically into three different types of steps. Okay, these are pre-treatment, saccharification, and fermentation. Okay, so pre-treatment is the you can say universally it is the first step when we carry out the conversion of the biomass into any useful product. Okay, because we have to reduce the size of the biomass, we have to make it amenable to the enzymes or the fermenting organisms. Okay, so for that purpose we carry out the pre-treatment, and it breaks the recalcitrant structure of the LCB by disrupting the lignin sheath. Okay, so the main target is the lignin reduction, but along with that we will also be able to remove hemicellulose. Okay, so that will make cellulose amenable to enzymes. Okay, so that we can you can say we can easily break down that cellulose into constituent sugar, that is glucose. Okay, so saccharification is the second step. It is the enzymatic degradation of pretreated biomass for the release of monomeric sugars like glucose. Okay. Then the last part is the fermentation, in which the glucose which is released from the saccharification step, it is converted into useful product. Okay, the example here is ethanol, but we can have variety of different products depending upon the type of organism that we use at this particular step. Okay, so this conversion of cellulose to glucose is carried out by certain enzymes. Okay, in the saccharification step, and those enzymes most in majority of the studies they are found to work best at 50 degrees centigrade. Okay, whereas the fermenting organism they work best at 30 degrees centigrade. Okay, so there is temperature mismatch, you can say, between these two steps. So it is always difficult to integrate these processes into single step. Okay, so we have to carry out these processes separately, or even if we are carrying out these processes in the same vessel, we have to maintain the conditions so that the enzymes and the yeast they work efficiently for their for conversion of the substrate into the desired product. Now, the pre-treatment process, so we have a variety of these different types of pre-treatment processes. We can categorize them into physical, chemical, physicochemical, as well as biological methods. So we can use different methods. These physical methods like mechanical processing, it is almost the universal first step in the processing of the biomass. Okay, so we have to first reduce the size of the biomass, then we have to apply different types of physical methods. Then we can also use different chemicals like dilute acid, dilute alkali. Okay, we can use them use them singly, or we can also use them in sequential manner or combined manner. Okay, so physicochemical processes are those in which we use the chemicals as well as some sort of other physical you can say energy. Okay, so we can apply high pressure, we can use high temperature. Okay, then we have the biological methods also, in which the fungi as such or their enzymes can be used. We can also use the bacterial or archaeal enzymes or the organisms to convert the to basically degrade the lignin that is present in the biomass. Okay, but these processes, biological processes specifically, they are comparatively slower and they are not preferred industrially. Although they are highly efficient, they are highly specific for the removal of lignin, but they are not industrially preferable. Okay, so we can have these combined methods where uh, like the advantages of two or more type of pre-treatment methods can be combined. So in the saccharification step, which is the second step, we use different types of enzymes like exoglucanase, endoglucanase, beta glucosidase. Okay, for the complete conversion of the biomass sugars into sugars. Okay, so the cellulose can be broken down by the chains of cellulose can be broken down randomly by endoglucanase enzyme, which is able to generate newer ends for other enzymes to work upon. Okay, so the cellulose chain, it will be attacked by endoglucanase, and this new chains will be generated, which will be shorter than the original cellulose chain. Okay, now these chains they can be acted upon by an exoglucanase enzyme, which can work on the reducing or the non-reducing ends. Okay, then beta glucosidase is the last enzyme in this sequence, which can convert the cellobiose and cello-oligosaccharides to glucose. Okay, so the final step is catalyzed by cellobiose enzyme, or which is also called as beta glucosidase. In addition to that, we can have the commercial preparations. Basically, they also have some other enzymes like hemicellulase, because some portion of the hemicellulose will also be there after, even after the pretreatment. We will not be able to completely remove hemicellulose. Okay, so to convert those remaining hemicellulose, we have especially in the commercial enzymes, we have the hemicellulase enzymes, different hemicellulases like xylanase, okay, mannase, and other type of hemicellulase enzymes. Then we can also have accessory enzymes, which are the basically the, you can say, oxidative enzymes, which can bring about the oxidative cleavage of the biomass. Okay, so overall these different enzymes, they work together to bring about the complete or near complete conversion of the biomass into respective sugars. Then coming to the conversion of the lignocellulose, the last step of this conversion, there is fermentation step. So we can have fermentation process we can carry out in different configurations that are listed here, like separate hydrolysis and fermentation, then simultaneous saccharification and fermentation, simultaneous saccharification and co-fermentation, and then consolidated bioprocessing. So these are the, like in literature, they are the established different types of configurations of the fermentation process that are possible. And most commonly these SHF and SSF process, they are commonly used industrially. Okay, although SSCF is also been tried, and CBB process, they are also being tried by different types of authors, but they are their commercial feasibility is still not very feasible. They are not commercially very feasible. Okay, so in fermentation process, this sugars, they are basically converted into the ethanol. Okay, and different variety of organisms can be used. Like we can use yeast, we can use different bacteria, which can be either you can say native type of microorganisms, or we can have genetically modified bacteria. Okay, so um, we have found that this SSF is found to be more economic and advantageous due to low production cost. Okay, because we can combine these two steps of saccharification and fermentation in the single process. So there will be like enzyme requirement and processing time, they will also be low. Then comparatively, it has been found that in comparison to the separate process, the yields and productivity of this SSF process is generally high, and it also overcomes certain inhibitions, like the inhibition due to the sugars that are there, because the organisms, they are constantly consuming the sugars that are generated in the hydrolysis process or the saccharification process. So this inhibition on the enzymes due to sugars, it is also not there, and it can operate under non-sterile conditions also. So this recalcitrance is one of the key limiting factor in the conversion of the biomass into any biorefinery product, because and because of this recalcitrance, so we have to work on that recalcitrance. We have to remove this recalcitrance, or this particular challenge during the conversion of the biomass, so we can improve the pre-treatment process. We can have new enzymes. Okay, we can discover new enzymes, or we can develop new enzymes, or we can develop the new capabilities in the enzyme, so that they can their efficiency is increased basically. And we can also improve the feed stock, because feed stock, if we can by genetic modification, we can reduce the content of the lignin, then it will be possible to maximize the conversion of the biomass into desired products. Okay, so I will skip this. These are the different properties of chemical composition and physical structure basically of the biomass. So it affects the conversion process in different manner. Okay, so I will not go into detail. So there are different components that contribute to these chemical composition of the biomass, like lignin, hemicellulose, acetyl groups, proteins. Okay, so these are the components of the biomass that contribute to the different chemical properties. Okay, and that may pose some inhibition on the conversion of the biomass into sugars. Then there are physical structure, or the properties that are related to the physical structure of the biomass, like crystallinity, then degree of polymerization, particle size, pore size, and surface area of the biomass. So we can change all these things with the help of efficient pretreatment process. Okay, so whenever we carry out the pretreatment process, it is expected that more than one of these properties will change. Okay, so ideally these properties should change when we carry out the pretreatment process. Okay, and along with that, the enzymatic digestibility of the pretreated biomass should also increase. Okay, so so in this manner, this pretreatment process is beneficial. Okay, because it will increase the accessibility of the cellulose. It will increase the enzymatic possibility of increasing the conversion is there. Okay, we can maximize the conversion of the biomass into constituent sugars. Okay, so this low accessibility of the enzymes is basically we can treat this by using optimal type of pretreatment process. Okay, and ultimately these whenever the enzymes they are accessible to the cellulose is accessible to the enzymes, then definitely this enzyme can work better, and it can bring about the maximum conversion of the biomass into desired product. Now I'm giving you a short you can say sort of exercise. Okay, so imagine that you are working in a you are running a factory basically. Okay, and there are different workers that are working to produce different products basically. Okay, it could be any industry, but for relevance you can correlate this factory with a biofuel industry, or you can say biorefinery industry. Okay, the workers will be yeast, okay, in this example, okay, or any other microorganisms that is going to convert the biomass into different components, and the product here is ethanol. Okay, so now just before I begin the discussion on like how these different types of inhibitors they are generated and how they are detrimental to the process, just consider this analogy that somehow the air in the industry it got toxic. Okay, some toxic gases are there, and the workers they are start slowing down. Okay, so they are getting sick, they are falling ill, and they are completely exhausted due to the presence of these toxic smoke, and the production is ultimately coming to a stop. Okay, so as an industrialist you should be trying to improve the productivity of the industry, you should be trying to increase the production. Okay, so what could be the possible scenarios? These are, although these are written here. Okay, so we can try to reduce the presence of this toxic smoke. Okay, or another strategy could be protective gears we are using. Why? Because we want to increase the capability of the organism or the workers here. Okay, so what this protective gear will do, it is not removing the inhibitors, or you can say the smoke that is present. Okay, but it is allowing the workers to withstand those type or work in that condition even if there's these inhibitors or the toxic gases they are present. Okay, so we can have different varieties of these strategies by which we can reduce the effect of these toxic gases, and we can increase the production of the industry. Okay, so we can improve the ventilation, we can install a smoke filter for continuous air filtration. Okay, or we can have a batch process, we can mix the fresh air into the factory so that the smoke gets diluted. Okay, we can move the factory to an area with better air quality also. Okay, then give workers protective gears, we can give energy drink so that their energy level is always high. Okay, or we can give workers a special medicine to detoxify this smoke into their bodies, or we can hire stronger workers also. Okay, that have the capability to resist this smoke. Okay, then we can change the factory design to reduce the smoke production. Okay. So I will come to all these solutions and this analogy once we complete this. We are towards the end of this lecture, so I will come back to this, and then we will see how this analogy works. We can correlate with this with the biofuel industry in terms of the inhibitors that are generated during the pre-treatment process. So this slide shows the origin of lignocellulose derived inhibitors. Okay, there are variety of these inhibitors, not one or two inhibitors that are generated. These can be generated in multiple manners, multiple processes. Okay, and in single step also, single process also, multiple type of these inhibitors they can be generated. Okay, so we can categorize for the sake of easiness. I have categorized them into four different categories. Okay, these are aliphatic acids like acetic acid, formic acid, and levulinic acid. Okay, then we have second category, which is the furans or the furan derivatives like furfural, hydroxymethyl furfural. Okay, then third category is the phenolics, which is the examples are vanillin, syringaldehyde, then ferulic acid. Then heavy metals and process residues. Heavy metals can be leached from the water which we are going to use in the biorefinery industry. It can be contaminated with heavy water, or there will be like during the chemical catalysis these inhibitors or heavy metals or process residues can be present. Then there are certain process specific factors also, like temperature, pH and time. Okay, so these type of compounds like aliphatic, furan, phenolics and heavy metals, they are generated during the process, pre-treatment process basically. Okay, but apart from that, we are also using some process parameters like temperature, pH, okay, and other factors like prolonged duration of the pre-treatment process. Okay, so these factors can also pose some inhibition on the enzyme. Okay, because high temperature, that temperature or pH might not be optimum for the enzyme to work. Okay, and then if we are keeping the enzyme or we are prolonging the time period of the hydrolysis process, then definitely the enzymes can get inhibited. Okay, so these different components, you can see cellulose, hemicellulose, lignin, they can give rise to these different types of products. So basically hemicelluloses, they give rise to when they are hydrolyzed during the especially when we are using the acid pretreatment process, dilute acid pretreatment process, or lignin removal during the delignification process, like we are using the alkaline pretreatment process. Okay, so these different inhibitors will be generated. So cellulose, hemicellulose, they can both be converted into oligosaccharides, which are the short chain you can say polymeric compounds, but they are very extremely short in nature. Okay, so these oligosaccharides, they also inhibit the enzyme. Then apart from that, there can be production of hexose sugars like glucose, mannose, galactose, rhamnose, then pentose sugars like xylose, arabinose. Okay, then when we are doing the pre-treatment process, so we are using the acid at high concentration. Okay, and we are also using the heat. Okay, so during these conditions, okay, these are the toxic conditions, so these sugars can be converted into different products, sugar derived inhibitors like furans. So HMF and furfural can be produced by the conversion of these different types of sugars. Okay, and later on in further presence of heat and acid, these furans can be converted into different aliphatic acids like levulinic acid, formic acid. Okay, and we can also have this acetic acid, which is present in this acetate group is acetyl group is basically present in the structure of the hemicellulose. Okay, so during the hydrolysis of those acetyl groups or removal of those acetyl groups during the pretreatment process, we can also get this acetic acid in the um, after the pretreatment process. Okay, then there can be uronic acids which can be derived from hemicellulose like galacturonic acid, glucuronic acid. Then we can also get the different phenolic compounds like coniferyl aldehyde, ferulic acid, 4-hydroxybenzoic acid, that basically comes from the lignin. Okay, so you see all these different types of components of the biomass, so they are the inherent, you can say, storehouse of these inhibitors. Okay, so whenever we are doing any pre-treatment process and the condition is very severe, okay, severe in the terms of these different factors, temperature, pH, time. Okay, so different combinations of these factors will ultimately lead to generation of these different type of inhibitors. Okay, so we have to play carefully with these physical parameters or physico-chemical parameters, you can say, acid or any catalyst that we are going to use. So we have to use it judiciously so that less of these inhibitors they are generated, and we can get more desirable changes in the biomass with respect to their you can say the bioconversion efficiency. Okay, so these thermal, acid or oxidative degradation of sugars and lignin produces toxic byproducts. So when we are going to use any type of pretreatment process, that will be based on one of these methods. Either we will be using heat alone or along with some catalyst, or we may use acid, or we may use oxidative process. So these will lead to basically degradation of the components of the biomass, and we will be able to generate that process will be able to generate these different types of inhibitors. Okay, so this is also similar to that slide. So these weak organic acids are acetic acid, formic, levulinic acid. Okay, these are derived from the acetyl groups in the hemicellulose and further breakdown of sugars. Then these furan derivatives, they can be formed from the pentoses and hexose sugars under heat and acid concentrations treatment. Then phenolics can be released from the lignin degradation during the pretreatment process. Okay, and heavy metals and process residues that can be due to contamination from the catalysts or reactor corrosion. Okay, so the fermenter or the reactor in which we are going to carry out the conversion process, that can also corrode, and it can lead to leaching of the heavy metals. Now, what is the mechanism of these inhibitions? So, what is the consequence of these inhibitors? So, ultimately, these inhibitors, when they are present in the hydrolysates, so they are going to affect the microbial, the microorganisms or the enzymes. Okay, so both enzymes and the microorganisms can be inhibited by these different inhibitors. So, different inhibitors, they work on different principles. Okay, there are different mechanisms by which these inhibitors can inhibit the microorganisms. Okay. So first, the weak acids, they basically disrupt the intracellular pH homeostasis. So intracellular pH of the microorganism, it will be changed. Okay, so that will lead to changed or the altered metabolism of the microorganism, and that will lead to impairing of the microbial fermentation process also, because the microorganism, it will not be able to carry out the its physiological process or biochemical cycles. They will be affected basically, and fermentation will be finally affected. Then furans, they are highly reactive. Okay, so they are rich in electron and they are highly reactive, so they will lead to oxidative stress. They can lead to oxidative stress, and they can also lead to DNA and protein damage. Okay, so the microorganisms, their macromolecular machineries, they will be affected by basically by presence of furans, and ultimately their growth and uh, multiplication will be ultimately affected. Then phenolics, they can disrupt cell membranes. They can cause leakage of the cytoplasmic contents, because these phenolics, they can be generated in different variety of these phenolics can be of the cell. Okay. Then heavy metals and process residues, they are also toxic to microbial metabolism, because they will affect the enzymatic activities of the microorganisms, and that will ultimately affect their physiology. Okay. Then apart from the fact the enzymatic processes, okay, these different types of inhibitors, they also affect the enzyme that we use during the saccharification or the hydrolysis process. So phenolics, they bind to the enzymes and they reduce their catalytic efficiency. Okay, so phenolics will affect the efficiency of the enzymes. Then furans, they will alter the protein structure, and they will ultimately affect the enzyme substrate interactions. Okay, so they will an acid can also cause the degradation of the enzyme. They can denature the enzyme, and they will further lower down the hydrolysis by the enzyme. And then heavy metals and process residues, they can also inhibit the enzyme activity in different manners. Okay, different heavy metals, they are toxic to enzymes also. Okay, they can affect different enzyme activities. So ultimately these different types of uh, like microorganisms or the enzymes, they will be basically affected. Okay, and overall the lignin, and those uh, lignin derived inhibitory compounds, they can cause the inhibition of the cellulase enzyme. Okay, which is the main enzyme during the biomass processing, because we are mostly interested in conversion of the cellulose into sugars, and for that cellulase enzyme is specifically used. So this lignin derived compounds can inhibit the hydrolysis process completely or significantly. Then this enzyme can also it can be blocked by the presence of lignin. Okay, the enzyme will not be able to proceed further in the cell. Okay, that can also hinder the activity of the lignin, the enzyme. So enzyme will not be able to enzyme will basically bind to this lignin. Okay, and but this lignin and cellulase interaction, it will be non-productive in terms of the sugar generation. Okay. Now we can also these different types of pretreatment process, they can also generate pseudolignin. Okay, pseudolignin is in some of the physical properties are generated from cellulose or hemicellulose. So these glucose and xylose, which are you can say the hydrolysis product of these uh, macromolecules, when they are converted into their different components like chlorfuel and hydroxymethyl furfural, which are the inhibitors. Okay, but they can directly act as an inhibitor, but they can also be converted into this pseudolignin, okay, through different chemical reactions. So these furfural and hydroxymethyl furfural are hard to remove. Okay, it will have altered chemical reactivity. It will also attach onto the biomass. Okay, it will get deposited into the biomass, basically the pseudolignin structure. Okay, one process. Now, what are the consequences of these LCB derived inhibitors? So the examples are shown here. So these inhibitors, they can get inside the cell of the yeast. Okay, through different transportation mechanism they can get into the cell. Okay, and they can affect the cell membrane or functioning of the cell, intracellular organelles. Okay, so there are variety of mechanisms by which the microorganism try to cope with these inhibitors. So when these inhibitors they are present, it is not like whenever these inhibitors are present, the microorganism will die instantly. Okay, there will be some long-term effect of these inhibitors onto the physiology of the microorganism. Okay, so once inside the cell, there are different mechanisms that the cell try to uh, uh, bring about. Okay, so the coping mechanism are efflux. Have some natural enzymes that can degrade these inhibitors. Then whenever these inhibitors they cause the damage to the cellular components like protein or nucleic acid or any other cellular component or macromolecule, then cells try to repair that damage. Okay, so ultimately what will happen, because all these processes, whether it is efflux or it is the detoxification, production of new enzymes, okay, or repair of the damage, so all these processes they will require some sort of energy. Okay, so basically what is happening, the microorganisms, in place of using the energy for the conversion of the sugars into ethanol, they are using that energy to cope. So ultimately what will happen if these microorganisms or the enzymes they are affected by these inhibitors? Okay, if any degree of inhibition is there, so ultimately the whole bioprocess will be affected. Okay, so these lignocellulose derived inhibitors they have detrimental effects on the biomass conversion overall, and reduced sugar fermentation efficiency will be there because there will be microbial toxicity. Okay, then the yield of ethanol will be low, means the biomass will be converted, it will not be fully converted into the desired end product. Then increase process cost, because all these strategies, for that we have to use some or the other type of detoxification method. Okay, so that will also lead to increase in the cost of the process. Then longer fermentation times leading to lower productivity. So overall productivity, because productivity is also important. Productivity is basically the rate by which the microorganism is converting the substrate into desired product. Okay, so definitely in the presence of these inhibitors, the microorganism will take more time to produce the desired product. Okay, even after that, the product will not be at the desired concentration. The concentration of the product will also be lower. Okay, so for this purpose, we should have like um, in the efficient type of lignocellulosic biorefinery, we should have strategies to mitigate these inhibitors. Okay, so we must have one or the other type of mitigation strategy by which we can mitigate the production of these inhibitors. Okay, so this table summarizes some of these different types of strategies that we can have. So we can first of all we can select the feed stock, okay, in such a manner that it has you can say minimal type of inhibitors are generated during the pretreatment process. So using less recalcitrant feedstocks and feedstocks that generate less inhibitors during the pretreatment. So definitely if the biomass is having less amount of lignin, okay, so lignin derived inhibitors can be controlled during the pretreatment process. And we can also use genetic engineering of the feedstock, genetic engineering of the crops, okay, through which we can modify the lignin synthesis pathway, okay, those pathways which are leading to the biosynthesis of the lignin. So we can reduce those enzymes, we can reduce those pathways so that ultimately the plants will contain less amount of lignin. Then we can use detoxification method, or we can use conditioning, okay. So detoxification method can be physical, chemical, or biological, okay. We can detoxification means we are chemically or physically or biologically converting those inhibitors into some other uninhibitory form, okay. So chemical additives can be used, like alkaline treatment we can do after the pre-treatment. Reducing agent can be used, and different types of polymers like polyethylene glycol can be used to tackle these type of inhibitors. Then we can also use bioabatement method, in which microbial treatment is applied, okay. So microorganisms, some of the microorganisms inherently they have the property to detoxify some of these inhibitors, okay. Or we can modify those organisms so that they can detoxify some new or the existing type of inhibitors. Then we can use culturing strategies like this SSF process, which is simultaneous saccharification and fermentation process. So this will basically decrease the feedback inhibition of sugars, and but we have to use large inoculum size, okay. So when we are using the fermentation with more quantity of the biomass or more numbers of the microbial cells, so definitely they will be able to resist, uh, um, they will be able to show more resistance towards these inhibitors, okay. Then we can select the microorganisms, so we can have different screening strategies for screening of these inhibitor tolerant microorganisms. uh, we can use evolutionary engineering method, which is the adaptive evolution using the specific inhibitors and lignocellulosic hydrolysates. So we can make the microorganism develop, um, you can say, in natural ways. The inhibitor tolerance will be generated through laboratory evolutionary laboratory engineering. Then we can also genetically or metabolically modify the microorganisms. Now coming back to this factory worker analogy, so we have already seen that these are the possible solutions. So we can now correlate how these different types of solutions they correlate with the real world biofuel mitigation strategies. Okay, so we can improve the ventilation so workers can breathe better. So detoxification methods can be used. They can be analogous to those ventilation strategy. Then we can install online detoxification methods like adsorbents. We can use biochar, okay. Biochar or activated carbon, it has the property to you can say adsorb some of the inhibitors, okay. Then we can use dilution strategy, so it is just like mixing the fresh air into the factory, so that in the slurry, okay. Then we can move the factory, so we can use feedstocks with lower lignin content to naturally reduce the inhibitor formation. Then protective gears are just like genetically modifying the microorganisms, okay. Then give workers energy drinks, so we can sort of we can compare with adaptive laboratory evolution to train yeast to grow better under stress conditions. So stress is being not being removed during this condition, but the microorganisms they are able to grow better even in the presence of inhibitors. Then we can introduce detoxification enzymes into the yeast through different um, um, engineering methods, okay. Like we can introduce aldehyde reductase to degrade furfural and HMF inside the cell itself. So once inside the cell, these inhibitors can be detoxified by the microorganisms. Then we can hire stronger workers, so we can develop, we can use robust non-Saccharomyces yeast, because mostly we use the Saccharomyces cerevisiae as the microbial chassis for the conversion of the sugar into ethanol. So we can use different types of these yeast which are non-Saccharomyces basically. Then we can change the factory, so we can optimize the pre-treatment conditions. So like we can use mild treatment, so that less of these inhibitors they are generated. Okay, so these are combination of these strategies, and no single strategy may work. We may have to use more than one strategy or a combination of these strategies to reduce the toxic effect of these inhibitors. Now coming to the third part, so I will briefly discuss about the results, what we have done in our laboratory. So we have used this adaptive laboratory evolution for mitigation of inhibitors, different types of inhibitors, both derived from the cellulose, hemicellulose, or from the lignin. Okay, so we have already published this paper in the Bioresource Technology journal. So in this work, we isolated and identified some thermotolerant yeasts. Okay, so initially we have seen that the yeast work better at 30 degrees centigrade, okay, in the mesophilic range. But for this study, we employed the thermotolerant yeast that are able to grow at temperatures beyond 40 degrees centigrade. Okay, and they are equally efficient. Even you can say they are more efficient at 40 to 45 degrees centigrade than at 30 degrees centigrade. So through this adaptive laboratory evolution technique, ALE technique, we have developed the resistance in the yeast strain towards these different inhibitor concentration. Okay, and we gradually increase the concentration of that inhibitor so that the yeast will be able to take up more inhibitors. It can grow better, it can produce more ethanol in the presence of inhibitors. So we don't have to basically remove the inhibitors from the after the pretreatment. We can directly use such a strain for the conversion of the pretreated biomass into ethanol. Okay. Then through optimization, process optimization, we have increased the concentration of sugar in the hydrolysate. We are able to more, you can say, produce more sugars, and we are able to convert those sugars into more ethanol, okay, by using this inhibitor tolerant yeast. And the same process has been validated at the fermenter level also. Okay, so I will briefly tell, like we have checked the, initially to design any strategy we have to set the benchmark, like from where we are going to start. Okay, so initially we did not know at what level the organisms they are able to tolerate the inhibitors. So first of all we checked that in the plate assay. Okay, so we grew these different strains. This is strain of the yeast, Pleuromyces margianus JKH5. So this yeast was grown in different concentrations of the inhibitors. So we took vanillin, furfural, and acetic acid as the representative inhibitors. Okay, and we saw that there were different these microorganisms. They were behaving differently to these different inhibitors. Okay, so because in the pre-treatment hydrolysate, these inhibitors they are present in the combined form, or they are all of these inhibitors they are present. So we also checked, we developed the cocktail of these inhibitors, 3 different inhibitors in different concentration, and we also checked the same in the broth assay. Okay, so we also checked the growth of the enzymes or growth of the organisms in the liquid broth. Okay. So initially, during these initial experiment, we were able to find that the cocktail of inhibitors, 3 gram per liter of acetic acid, and then 0.1 and 0.1 gram per liter of furfural and vanillin, these were easily tolerated by the microorganism. Okay, so this one. So we chose this as the benchmark, and we initiated our study using this concentration, and then we finally gradually increase the concentration of these inhibitors, and we grew the culture in the continuous presence of these inhibitors. Okay, so we use this strategy. So the culture was transferred to the broth. Okay, and then we grew it for certain generations, like 10 to 20 generations in the same concentration. And when we found that after 12 to 14 hours of the growth, or maybe 16, 18 hours, the organism was able to grow properly, then we checked out the growth profile, and we compared it with the parent enzyme, parent organism. Okay, so wild type yeast, we compared it with the wild type yeast, and whenever there was any significant improvement in their growth behavior, so we moved on to the next concentration of the inhibitor. So we increase the concentration, and then we again try to adapt that cells into that concentration, and then we again checked those properties, different parameters of the growth, and whenever there was significant improvement, so we increased further. So in total, we performed this experiment for approximately 60 to 70 cycles, okay, in the presence of inhibitor. So we compared, uh, because initially we did not know like how to proceed, okay. So we devised several strategies, like one was to grow the organisms into different inhibitors individually and then shift to some other type of inhibitor. And one strategy parallelly we applied, like using the cocktail and increasing the concentration of the inhibitors in the cocktail of the inhibitors, okay. So we later on we found that at similar type of generations, like 60 to 70 generations of all these inhibitors, we were able to increase the tolerance of the yeast, okay. So in case of acetic acid, furfural or vanillin, we have to take 3 or more set of experiments to generate the uh, tolerance towards another type of inhibitor. So we took this yeast which was tolerating the cocktail of inhibitors, which was adapted to the increased concentration of the cocktail of inhibitors, and we found that it had good growth characteristics, like the specific growth rate and the doubling time of this uh, uh, yeast that was uh, able to resist different types of cocktail of the inhibitors. So it has better growth, okay, which was evident from the doubling time, decrease in the doubling time, and increase in the specific growth rate in the presence of inhibitors. So thereafter we also checked the and compared the ethanol production by this particular strain, okay, and we compared it with the parent strain. Definitely we got significant improvement when the same strain was grown in the presence of inhibitors, okay. So it was tolerating the inhibitors, and at the same time it was utilizing the sugars for the conversion of sugars into ethanol, okay. So that concentration of the ethanol was also good. And later on we used this sequentially treated biomass, okay. So biomass that we took was sugarcane bagasse, and sequential pretreatment was dilute acid and alkali pretreatment. Through pretreatment we were able to increase the cellulose concentration to around 83%, okay. So this was a two-step pretreatment process or combined pretreatment process, and we performed the separate hydrolysis of this sugarcane bagasse. Okay, so in separate hydrolysis, first of all, we carry out the hydrolysis process, and then we carry out the fermentation of the released sugars. Okay, so even in presence or presence of inhibitors, there was no significant decrease in the production of the ethanol. Okay, so almost you can see the ethanol concentration in the presence of inhibitors or in the absence of inhibitors, it was almost similar. Okay, so later on we moved on to the fat batch process, in which we adopted two different strategies, okay. So fat batch is basically the process in which we apply the substrate into different batches, okay. We don't directly apply because the sugars or the inhibitors that are present, they may the organisms may not be able to convert them efficiently. So we tried this fat batch approach, and we tried two different approaches. One was to feed the biomass alone at different time intervals, like at 6 hours. So initially 1/3 we had at the 0 hour, then 6 hours, then 12 hours, and then we proceeded with the fermentation process. Then second strategy was to feed the biomass along with the enzyme, okay. Because whenever we are going to produce ethanol, so we also need to increase the concentration of the ethanol, okay. We always desire that we get high concentration of the ethanol, okay. So in any biochemical or chemical reaction, uh, what is the like how you can increase the concentration of the product? How you can increase, if you want to increase any concentration of any product, so what you have to do basically? You have to increase the concentration of substrate, okay. Although it may not be directly correlated, but one possible solution could be to increase the concentration of the initial substrate, okay. But the limitation with ethanol or bioethanol is that we cannot increase the concentration of the biomass up to after a certain interval, okay. So 5% or 10% of the solid biomass can be efficiently used, but with that biomass, even if it is containing 100% cellulose and 100% cellulose is being converted into 100% glucose, even then we will not be able to achieve high concentration of the ethanol. So we have to increase the concentration of the biomass in the initial, you can say, in the reactor itself. Okay, so for that purpose we try to increase the concentration. The main purpose of this fed batch process was to increase the amount of the biomass in the reactor. So ultimately we got good results with this 20% solid loading. Okay, and with feeding of biomass as well as enzyme at different time intervals. Okay, so we could get around 80 gram per liters of ethanol. Okay, and that is a good amount considering the industrial production of ethanol, or even if you consider like using the thermotolerant yeast, because the yeast is working at 40 or 42 degrees centigrade. Okay, so that is good amount of ethanol that is being produced. So the maximum ethanol was 85 gram per liter. It was obtained during the fed batch mode of the fermentation. So these were the experiments that were conducted at the shake flask, and then we moved on to the fermenter level also. Okay, and we could achieve good amount of ethanol production. So in that case, uh, if inhibitors uh, were absent in the this fermenter, so definitely the ethanol production was better or more, 82 gram per liter. But even in absence of in presence of these inhibitors, the yeast was doing fairly good, and it could produce 73.4 gram per liters of the ethanol with yield of 0.44 and 2.6 gram per liter per hour of the productivity. Okay, so industrially these are very relevant because we need high, all these parameters must be high. This ethanol concentration as well as the yield and the productivity, they should also be very high. Okay, and we cross the benchmark for the industrial, you can say industrially feasible ethanol concentration is 40 grams per liter. So we were far away from that concentration, and this process was successfully demonstrated at the 3-liter fermenter level. Okay, so we could get maximum of this 82 gram per liters of the ethanol. Okay, so we also did this mass balance analysis, and this mass balance analysis revealed that out of the total initial 1 kg of the biomass, we could get 260 grams of ethanol. This data based on, and you can also while applying, uh, so these were the results. So while applying this concept of inhibitors, okay, and you are applying the LC analysis, okay, some of you might be interested. So you can also take into account these inhibitors as one of the critical parameters, because although these are the inhibitors from bioconversion point of view, okay, but some of the inhibitors like furfural and HMF, they are very useful for chemical industry. Okay, but the main thing is separation from the hydrolysate. So after pretreatment we have to remove these inhibitors, but still they have very high commercial value and they can be used as a starting material for many chemical reactions. Okay, so it is our point of view, like from bioconversion point of view, these are the inhibitors, but for chemical industry they can be the further substrates. Okay, so if you apply LCA and if you apply the cost benefit analysis, so your perspective may lead to different results. Okay, so you have to decide whether you are considering these as a waste or you are considering these as a further, you can say, useful chemicals. Okay, so with this I conclude, and the conclusions are this: lignocellulose derived inhibitors, they are the major challenge in the biomass conversion, and understanding their formation, inhibitory effects and mitigation strategy is crucial for optimizing the biorefineries. And combining the pretreatment optimization, microbial engineering and detoxification strategies can enhance the biofuel production efficiency. And one of the mitigation strategy like adaptive evolution has proven successful in improving the cellulosic ethanol production in the presence of inhibitors. Okay. So I acknowledge the funding agencies, different funding agencies, CUH, then lab members, students, collaborators and mentors. Okay. Thank you. So if you have any questions, you can ask. Outside participants. No questions?
Sir, what could be the possible mechanism through which these inhibitors, through which these microbes can get to adapt to these inhibitors?
The main mechanism is like the genetics of the microorganism, because the spontaneous mutation they are going on in any organism, in each and every generation. So they are random mutations. But what we are doing basically during the adaptive evolution process, we are increasing the speed of those uh, uh, mutations basically. And after that, we are selecting the microorganism. We are keeping some sort of selection pressure, continuous selection pressure, that is the presence of inhibitors. So the microorganism has to if it has to survive, so it has to develop some sort of mechanism, but that mutations can be at different regions, okay, different mechanism, and more than one gene may be mutated. So ultimately we have to compare and see like what are the genetic changes that have been there in the adapted organisms in comparison to the parent. Only then we can specifically say that this particular type of mutation or change has been taken place.
So can we say that it is a directed random mutation?
Yeah, that's why this is adaptive laboratory evolution. Evolution is there, evolution term, but we are carrying out in the laboratory in the directive manner, because we are forcing the microorganism to go through those harsh conditions, okay, that will make it stronger, and that will make it to adapt some strategies to survive under those conditions. Okay, so we are forcing it basically. Yeah. Any other?
Sir, can we introduce some bacteria or fungi to uh, adopt inhibitors or adopt or utilize the inhibitors that may restrict the reaction during the our biophenol production by genetic engineering?
You mean to say we can introduce at what step?
Uh, the inhibitors which are produced, some bacteria or fungi we may develop by genetic engineering to utilize them in the same process.
Yeah, we can utilize, we can develop some organism, but for that again we have to do some targeted engineering. We have to target certain genes, and in most of the cases, because whenever we are talking about any inhibitors in general, so there are lot of these inhibitors.
Yeah.
So you have to have microorganism which is engineered to uh, take up all those different types of inhibitors and convert those into or neutralize those.
Sir, we
Okay.
can also do hybridization method, which some of them are separated from the our process, and some of them can be utilized by the microbes.
Yeah, we can do, but that will you can say that will increase the efforts. More efforts will be needed, because we have to develop different strategies to tackle all these inhibitors, because different types of inhibitors are present, not single one.
Yes.
So we have to make the microorganism compatible to all different types of inhibitors. And then again, we are not only going to use single type of process. Okay, there are different multiple pretreatment methods. That whenever you are changing the substrate, you are changing the fermentation strategy, you are changing the product out of the biomass, so you have to use different type of pretreatment method, and
Yes.
that may lead to differential production of these inhibitors. So it is not like like in our example we have treated sugarcane bagasse basically.
Okay. Sir.
Okay, but if we change the biomass, then definitely the inhibitors will be different. So that organism may not be able to, it may survive, it may convert those inhibitors, but definitely there will be variability in its functioning.
Okay, sir.
Okay.
Thank you, sir.
So that will be a challenge. So no questions. So we can close this session.
Thank you so much, sir.
Thank you.